So you say to me, what additional implications might this hold? Okay? And how can we actually interpret this in terms of specificity? Okay? Well, one of the things that you can think about, what this might suggest, is that if you review the literature, in the literature, RNA transcribed in the nucleus can actually exit the nucleus into the cytoplasm through one of two major pathways. Okay? One is going through the CREM transport pathway, and the second pathway is through the TAP transport pathway. And there is a hypothesis in the literature that in fact suggests that M7G-CAP transcripts go through the TAP pathway, and TMG-CAP transcripts go through the CREM1 pathway. Now, of course, those of us working on HIV transcription always thought that, in fact, the single determinant of whether viral transcripts go through the CREM1 pathway or not is actually the Rev-RRA interaction. But here, in fact, we have the evidence that, in fact, the determinant may not actually be Rev-RRE, but this may actually be whether that particular transcript, the HIV transcript form, is M7G-CAP or TMG-CAP. Okay? So you say, well, how could you possibly link those two sort of unrelated observations together? And I think you can link those two observations together by asking the question that in the case of HIV, where we have a form of HIV RNA, which is expressed from the GAG-POL-RRE form, which is Rev-dependent, versus a form of the RNA, which is expressed from GAG-POL-CTE vector, which is Rev-independent. Okay? So here it goes through the CREM1 pathway, and here it goes through the TAP pathway. Okay? Which one of these two transcripts that are in fact identical, okay, in their transcript form, except for the RRE sequence versus the CTE sequence, and in the presence of Rev, which one is PIMT dependent and which one is PIMT independent? So it turns out that this form is PIMT dependent, and this form, if you express the transcript, is in fact PIMT independent. Okay? So how does that fit then? How it fits is because it turns out that PIMT is a factor that directly binds to the Rev protein, and it is recruited to RRE-containing transcripts by Rev. And so the model that we believe is happening is that transcripts are made, Rev recognizes the RRE, the transcripts are POL2 transcripts, so they go through the default capping, which is the M7G, but the Rev then recruits PIMT, which then hypermethylates this into the trimethylated cap, and in the trimethylated cap form, this is then recognized by CREM1, and then it dictates the transcript of Rev-RRE dependent transcripts through the CREM1 pathway. These transcripts that are singular, just single methyl cap, in fact can go through the TAP pathway or go through some other pathway. But in order to go through the CREM1 pathway, you have to have the trimethylated cap and the previously demonstrated essentiality of Rev-RRE interaction is purely for the purposes of bringing a trimethyl transferase here to trimethylate the cap. And it is actually the identity of the cap that determines the transport through this particular pathway. And this would, in fact, agree with the literature on cellular RNAs, where two different forms of cellular RNAs, the trimethylated form of cellular RNAs, are generally transported through the CREM pathway, while the monomethylated cap, our cellular RNAs, are transported through the TAP pathway. Okay? So now, if this is in fact true, if this model is in fact true, then we can predict three findings that we should see. One is that we should see that PIMT should increase the trimethylated cap on Rev-RRE transcripts, but not the trimethylated capping on the same HIV transcript, that is GAG-POL-CTE. Okay, so GAG-POL-RE should have trimethylation increased by PIMT. The second prediction is that when you do an immunoprecipitation of the PIMT protein, you should only be able to pull down the GAG RRE containing transcript, and you should not be able to pull down the GAG Paul transcript that contains the CTE, and that is also the case. And finally, if you look at the transport profile of PIMT, you should see that it should exhibit leptomycin B sensitivity, as we would predict if this particular protein is going through the CREN1 transport pathway, as opposed to some other way of shuttling between the nucleus and the cytoplasm. Okay? And indeed, this is the third prediction that is also verified. So when we have no LMB, you find the PIMT can distribute from Cajal bodies and into the cytoplasm. So you can see that's a nucleus in blue. Then if you do LMB treatment, then you find the PIMT is all sequestered in the nucleus, and it's still increased in the Cajal bodies as we would expect, and this is indeed what we will see. So the predictions then all agree that in fact, this particular factor is an important factor that regulates the modification of CAPs on HIV. And ultimately, it's the modification of CAP that determines the fate of the transport. It is not actually REV-RRE. REV-RRE is there to change the modification of CAP. And this, in fact, becomes a unifying finding that explains how cellular transcripts are also discriminating between different pathways, and HIV is doing exactly the same thing. So my time is up. I skipped the second half, which is talking about RNA helicases, which we have now characterized several additional ones for post-transcriptional expression of HIV, and simply go to the summary of the 2 points, which is that the cap structure is a determinant of the trafficking pathway, and it's a cap structure of HIV transcript that ultimately determines the specificity of expression. And finally, to the most important slide, which is to acknowledge the postdoctoral fellows in my lab and collaborators who work on this and other projects with us in our lab. Thank you very much.
Questions, Simon?
Yes, Simon.
Simple question. What's in the virus?
What's in the virus?
Is it trimethylated or mono?
It's a good question. It's a good question. We don't know. We don't know. We've thought about that question too, but we just have, this is really fairly new, and we just have not looked at that.
Maybe. Yeah.
Could you comment on the implications of the trimethyl cap for cap binding protein or interactions with the decapping complex?
Don't know. Haven't looked at it. Sorry.
If here, this side, if your model is correct, you would expect that tethering that directly the PIMT protein to the RNA should make the RNA REV independent. So did you try this experiment? Like with MS2? Or...
I think if we bring REV to an RNA, so if we bring REV to an RNA, our prediction is that it will then bring PIMT to the RNA and then trimethylate the cap.
Right.
And I think that's an easier way to do it, to make a MS2 REV fusion. And
no, but if you would do a PIMT MS2 fusion.
Okay, yes. That's
directly,
also,
just to shunt completely the REV.
Right, right. No, yeah, we haven't done that, but yes, it's a good idea.
Team, I have a small question. It has been shown in alpha retroviruses like RSV that you have also methylation within the leader sequence. Have you looked at that?
Methylation in the leader sequence.
Yes, internal methylation.
Well,
what the status with respect to HIV?
Yeah, we would love to do that, but you see, we're limited by, we could do these experiments because there are very good antibodies to discriminate between the different forms of CAP. If you have very good antibodies to discriminate that, then we could do that experiment.
Well, last question.
I have a question. I would like to know which region of REV is